In this video, we'll continue about the biology of Leucosolena. In the previous video, we have talked about the characteristic of sponges, and we have already said the characteristic of sponges is that they are multicellular and they have a versatile cells that is capable of what? That's capable of division of labor, and also they are what? They appear in different shape, size, and color, and they are found attached to a substratum. They are found attached to a substratum, and the substratum is what? Is an object like seaweed, like the one I'm showing right now. Look at it. This is an example of sponges. You understand? This is an example of sponges. Now we said they have pores all over their body. Look at their body, they have pores all over their body, and their pore is termed the ostia. You understand? The pores is termed the ostia, and the ostia is produced by what we know as the porocytes. And we say they have three layers. They have the inner layer, the outer layer, and the middle layer. And the inner layer is termed the pinacocytes. The middle layer is termed the mesohyl, and the outer layer is termed the pinacocytes. The middle layer is termed the mesohyl, and the inner layer is termed the what? The choanocytes or the choanodam. So we'll continue from where we stopped in the last video, and that is what? That is nutrition in sponges. Now talking about the nutrition in sponges. Now, like I said, all sponges are suspension feeders, and they feed on food that are suspended on flowing water that contains fine materials. And how do they feed? Water that is flowing through the osculum. You remember I told you that the osculum serves as the mouth. You understand? By the beating of their flagella. Once they beat their flagella, they keep beating their flagella. Once they beat their flagella, it is going to cause turbulence in water. Now, it is going to pass in food, suspended food, in what? Inside water into their osculum. Are you getting that? It passes what? It passes suspended food into their osculum. Now, once it passes suspended food into the osculum takes it in. You understand? The osculum takes it in into the what? Into the spongocoel. To the spongocoel, and this food particle is then phagocytosized and digested intracellularly. Please note that it is digested what? Intracellularly. So they undergo what? Intracellular digestion. I think I wrote it somewhere around there. Yeah, intracellularly. Is it? Intracellularly. I get me now. Now they now have they have numerous cytoplasmic extension, and this cytoplasmic extension is termed microgyl. You understand? It is termed microgyl, and this microgyl form mesh of network which filters this particle. You understand? It filters the particle that is needed by the body. It filters it from those one that is not needed. You understand? Then after it filters it, the food particle is then ingested by the cell body of the choanocyte and digested intracellularly. Once it is digested, it is now transferred to the amoebocyte. Are you getting me now? Now, the amoebocyte now take it into the what? Into the mesohyl. From the mesohyl to other cells. You understand? Other cells of the sponge body. While the water and the undigested particle passes out through the osculum. You understand? Through the what? Through the osculum. Are you getting that? So that is what? That is the osculum also serves as the anus. Are you getting that? The osculum also serves as what? As the anus. So that is how they feed. And they store food as what? They store food as lipoprotein or glycogen. Now, how do they respire? They do not have distinct organ for respiration and elimination of metabolic waste. You understand? Hence, the gaseous exchange and discharge of waste products is achieved by the process of simple diffusion. It is achieved by the what? By the process of simple diffusion between individual cells and water flowing through the body. And they excrete in form of nitrogenous waste. You understand? A nitrogenous waste in form of ammonia. That is how they excrete. Now, what about their reproduction? They exhibit two types of reproduction. They exhibit sexual and asexual reproduction. They exhibit what? Sexual and asexual reproduction. So let me write sexual. Okay, I don't need to write that. I think I put it here. We have either it is sexual reproduction or asexual reproduction. Now, how do they undergo sexual reproduction? sorry, asexual. Asexual is by budding. Now they have two type of bud. They have what? Two type of bud. Either internal bud or external bud. You understand? Either what? Internal bud or external bud. And this internal bud, if it is internal, it is called gemmus. Gemmus. You understand? But an external bud is most common. It is the most common bud that they actually form. They usually form. You understand? Now let me just illustrate that. Let me illustrate the budding. Now, let's just check this. We have a what? We have a particular this thing. We have a particular Leucosolena. Remember, they are tube-like. They look like a tube that is attaching to a substratum. You understand? Now, since they attach to a substratum, this is a matured Leucosolena. Now wants to undergo asexual reproduction. Now, it is now forming a what? Another this thing. Then it will form something like a bud outside. Can you see? Form something like a bud outside, still attaching to the what? Still attaching to the substratum. You understand? But this one that I marked in what? In pink is called what? Is called the bud. You understand? That is the bud. So then it is now moved to a mature bud. The Leucosolena is now going to have a mature bud. Can you see? The bud is now matured. The bud is now matured. Ready to what? Ready to be separated. Then it is now going to separate into two Leucosolena. You understand? Two of them. That is what? That is the burden of what? That is the burden of these animals. You understand? So this is just an illustration. This is not really how it looks like, but me, I'm just trying to like illustrate it. You understand that? So that is how it is illustrated. So we have the daughter in Leucosolena and the what and the this thing. Remember, it has an opening around there, and the opening is called osculum, which serves as mouth and the anus. You understand? So look at this is the structure of Porifera. Look at it. Remember, I told you they have the outer cell, and the outer is called the pinacocyte. They have the middle called the mesohyl, and they have the inner called the what? Called the choanocyte. You understand? Now, these amoebocytes, I don't know if they put it here. I don't know. They did not put it here. Okay. They have a cell called amoebocytes. Amoebocyte is called jack of all trade. Amoebocyte. Amoebocyte is called jack of all trade, and it is also called wandering cells, something that wander around, something that over around. You understand? Because it performs almost all the function inside the animal. You understand? So it is one of the most important cell in the what? In the body of that sponge, in the body of a sponge. You understand? Remember that digestion is what? Intracellular. So these are the what? These are the three cells that is inside their what? That's inside their body. Now you have to note that they also have contractile vacuole, and this contractile vacuole is used for osmoregulation. And let me see if I can actually see that here. No, I can't. Let me add that one. So they have what? They have contractile vacuole inside the what? Inside the amoebocytes. Amoebocytes. And this contractile vacuole is used for what? Is used for um osmoregulation. You understand? It's used for what? Osmoregulation. Are you getting that? Now, I've talked about the what? I've talked about the asexual reproduction, Abby. So I'm going to talk about the sexual reproduction very soon. So this is how they look like. Can you see? This is how it looks like. This is how the sponge. This is how this thing looks like. Can you see? This is their flagellum. Eh? This is their what? This is their prosospile. Prosospile. This is water flows in and water flows out. You understand? Water flows in and water flows out. Now, this is how it looks like. So look at another structure of them. Look at the another structure. So look at Leucosolena. This is how a Leucosolena looks like. Sorry, I have to cancel this. This is how a Leucosolena looks like. Can you see? This is how it looks like. So if you notice very well, it is having a bud somewhere around there. Can you see the bud? So the bud now separates to form a new Leucosolena. Can you see? They look like a tube. They look like a tube, and this is their structure. So they have the what? They have the outer pinacocyte. They have the outer pinacocyte. They have the middle mesohyl, and they have the inner choanocyte. So these are the what they possess. Now, talking about the next one, that is their sexual reproduction. Now, the sexual reproduction is quite interesting. You understand? Now, they shed sperm and egg into the sea. Sperm and what? Sorry, they shed only sperm into the sea. Just the what? Just the sperm into the sea. Now, the amoebocyte, remember I told you the amoebocyte is the what? Amoebocyte is the wandering cell. The amoebocyte now prevents the amoebocyte prevents the maturation of egg and sperm at the same time, which means because these animal are hermaphrodites, they have what? They have the male and female what? The hermaphrodites. Hermaphrodites. So they have the male and female organelles in the same animals. You understand? So because they are hermaphrodites, they don't want self-fertilization. They don't want to undergo self-fertilization. So the egg and the sperm is going to mature at different time. You understand? So to prevent what? To prevent self-fertilization. That is why they mature at what? At different time. Now, once the sperm is shed out into the sea, it swims. When it swims, it is taken into the mesohyl, that is the middle layer of another sponge. Then it fertilizes the egg, and the egg is termed the oocyte. The egg is termed what? The oocyte. Now, the choanocyte now transfer itself into the into transport what? Into transport cell, which transfer it to the what? To fertilize the oocyte. Once it fertilizes the oocyte, it is going to form a zygote, and the zygote undergoes holoblastic cleavage. Holoblastic, let me add another page. Holoblastic what? Holoblastic holoblastic cleavage. And once it undergoes holoblastic cleavage, it's going to form what we know as blastula. Blastula. Now, this blastula now develops. The blastula develops to form what we know as stomoblastula. From what? From blastula to what? To stomoblastula. Now, the stomoblastula in the inner part, it consists of what? Non-flagellated cells and flagellated cells. Now, the non-flagellated cells now invaginate inward, and the flagellated cells invaginate as well to form the non-flagellated cells form the posterior pole, and the flagellated cells form the what? Forms the anterior pole. Now, once they do this, they now develop into what we know as, remember, from blastula to what? To stomoblastula. Then the stomoblastula now develops to amphiblastula. The stomoblastula develops into what? Into amphiblastula. So these are the what? These are how they reproduce. You understand? So once they develop into amphiblastula, amphiblastula is now the what? Is now a flagellated swimming larva. The amphiblastula consists of flagella. You understand? Something like this that consists of flagella. So it consists of flagella all that, but so it swims. Once it swims for a while, then it gets itself attached to a substratum. So we are going to have something like this attached to a substratum. So it's going to lose its what? Its flagella. You understand? It's going to lose its flagella, and it's going to form what? A matured sponges. You understand? A mature sponge. So the amphiblastula is what? Is the larva of sponges, and it is what? It is free swimming. It can swim. You understand? So what have we talked about so far? We talked about their nutrition, and we said they are suspension feeders. And from there, we mentioned that the water enters through the osculum, and it is taken into the spongo coil, from the spongo coil to the what? To the amoebocyte, to be digested intracellularly. Then the nutrient is now moved around by the amoebocyte to the mesohyl, to other cells of the body, and the undigested particle is taken back into the osculum. I'll mention their respiration. Respiration is by simple diffusion, and excretion is also by simple diffusion. And they have something called nitrogenous waste that is in form of ammonia, right? And we talk about their reproduction. I said they can be reproduced both sexually and what? And asexually. Both sexually and asexually. So this is my own illustration of these their layers. So they have three layers. Look at. I'm sorry for my handwriting. Handwriting is actually very, very beautiful. So so this is the what? This is they have the outer layer, the inner layer, and the what? And the middle layer. So we have the choanodam, we have the mesohyl, and we have the what? We have the porocyte. So this is just it. You understand? This is just my illustration. Now, I said they can undergo reproduction, either sexual or what? Or asexual reproduction. And the sexual reproduction is by what? Is by fusion of sperm and egg, which is called the oocyte. And the asexual reproduction is by what? Is by budding, like formation of bud. And I showed you something like this. I showed you Leucosolena. So this is a typical example of what? A typical example of the sponge. You understand? So this is how the phagocytosize food to the what? To the choanocyte, to the amoebocyte, then all over the body. And they beat their flagellum. Remember, they always beat their flagellum. And that is all about the phylum Porifera. Thank you for watching and stay tuned for the next video, which is based on the phylum Cnidaria, where we talk about the hydrozoan, the anthozoan, the scyphozoan, the cobozoan, and even the sea animal called the actinia. Thank you so much.